Hi, everyone. I'm Clement Coclet. I'm a postdoc working at the GGI. And today I'm going to talk about biodiversity and activity associated with seasonal turnover in the mountainous watershed soil ecosystem. So as you probably already know, climate change impacts natural ecosystem all around the world, and especially the mountainous ecosystems. With reducing the snow melt cover and the snow cover, and also trigger an early snow melt. And all of this climatic change have significant impact on the soil microbial communities and their biogeochemical cycling associated with. So more and more study about viruses on DNA and RNA sequencing have been proposed. And all of these studies show that the soil virusphere are very diverse, abundant, and they can infect a large range of microbial hosts. And they also can affect the biogeochemical processes, especially in soils. However, there is a lack of knowledge about the soil biomix, and it is why we need more research efforts on soil viral ecology, especially in mountainous environments. And it is why I'm here today to present you a study what we have done in the East River Watershed in Colorado. This site has been chosen as a representative mountainous soil ecosystem to study specifically the climate change on the microbial community and the viral community. So this representative mountainous site allows us to study the diversity of both DNA and RNA viral communities. More specifically, I will focus on phages today. We can also investigate the phage-host relationships and the nature of this interaction in soils. And we finally also studied the activity of both DNA and RNA phages and the genes they carry in their genomes. And finally, our long-term goal is to add this viral component on a more global system of microbial dynamics and ecosystem functioning, especially related with the climate change. So how to do that? First, we collect some soils from September to June in order to study the viruses at different seasonal conditions. We also collected samples at 3 different depths and at 2 different at 2 different locations in the watershed. So this sampling gave us 48 metagenomes and 44 metatranscriptomes. And on these samples, we applied multiple bioinformatics tools to identify viral conjugates in these samples. And on these viral conjugates, we run multiple downstream analyses to study the diversity of these viral conjugates, to study also the viral-host interaction, and also the activity of these viruses. And at the end of the day, we had this very unique data set, which allow us to study the dynamics, the diversity, and also the activity of the mountainous soil DNA and RNA viral communities. So we identified more than 4,000 and 5,000 DNA and RNA viral community, respectively. Regarding the RNA viral community, it was a very large and unexpected number of RNA viruses. So then we wanted to understand or investigate how these communities were driven by the environmental conditions. So here you can see 2 NMDS of showing you the distribution of metagenome and metatranscriptome samples through the 4 sampling months. So to the left, it is the GNA1 and the RNA1. And as you can see, both communities, so both DNA and RNA communities, were exhibited some seasonal variations with the March and September was, the March month was very apart compared to the 3 other months. On the contrary, we didn't find any variation by depth or sampling site. And here you can see a box plot showing you the Bray-Curtis dissimilarity between the closely relative month, with in blue the value for the DNA1 and in green the Bray-Curtis value for the RNA community. And a high Bray-Curtis dissimilarity means strong differences in terms of RPKM abundance between the 2 compared months, so between the pair of months. And so what you can see here is that for each month comparison, the Bray-Curtis dissimilarity were higher for the RNA viral community than for the DNA viral communities. Which means that the RNA viral communities seems to be more dynamic than the DNA one. And the DNA viral community appear relatively stable over months. So after that, we wanted to investigate the DNA viral community composition. So because the DNA viral contigs were predicted from metagenomes, and based on their gene content, we estimate that the majority of the identified DNA viral contigs were phages. And because they were phages, we were able to run a new tool developed by Simon called IFOP or IPOP, I don't know, to predict their microbial host. So here you can see a bar plot showing you the relative abundance of the DNA phages colored by their predictive host. And not very surprisingly, the majority of the phages, of the DNA phages, infected actinobacteria and alpha proteobacteria, which is a common bacterial group in soils, in soil microbiomes. However, we found some phages that infected specific family of bacteria at very specific month. So for example, here you have some phages we infected the Actinomyceta group only in September, and so on and so on. So we saw a seasonal turnover of DNA phages based on the host dynamics in soil. And this dynamics that we observed was very consistent with the bacterial turnover observed in soil on the previous study based done by the team we gave us the samples in Berkeley Lab. So then after that, we also wanted to investigate the RNA viral composition. So to do that, we perform a phylogenetic analysis based on the gene encoding for the RGRP, which is a gene found in each RNA viruses, and it's kind of an homology with the 6nes RNA gene for prokaryotes. So the RNA viral community was largely dominated by the single-stranded RNA Lenarvieri-Cotta, and with a significant fraction of Levi-Ricetes, which are phages, so viruses infecting bacteria. Here you can see an example of a tree we have done. So we did a phylogenetic tree for the 5 different phylum. So here is an example of the tree for the phylum Lenarvieri-Cotta. So in green, the green and the red branches correspond to newly and previously observed viral cluster, respectively. So in green, this is cluster with only viral contigs from our study, and in red, this is cluster with contigs from our study and contigs from other studies used as references. The first layer shows you where each viral cluster are from. So based on the reference study we include in this phylogenetic analysis. While the second layer represents the predictive analysis, the predictive taxonomy, sorry, based on the RefSeq database. So we identified a lot of new viral cluster in green, which are potentially new RNA phages or viruses. Then we also wanted to investigate the virus-host relationships, and with this phylogenetic analysis and using the RefSeq database, we were able to predict the putative host for at least most of the viral cluster in this tree. So as you can see, there is a lot of Lennar vericota associated with bacteria here in purple, which is phages. We also find a significant number of viral clusters associated with fungi, and some of them associated with invertebrate hosts. And finally, we also wanted to investigate the activity of these buff communities. So for the DNA viruses, basically it's more simple than for the RNA. For example, for a given DNA viral contig, if at least one gene were found in both metagenome and metatranscriptomes, we classified these contigs as active. For the RNA viruses is a little bit more tricky because we cannot compare metagenomes and metatranscriptomes. So we only worked on RNA viruses, single-stranded RNA viruses, because they had only one strand. And if we found both strand in the metatranscriptomes, we assume this viruses is doing a replication cycle. So we classify these contigs as active. So here we go, it doesn't work. Same one is me. So no, it's my PDF. Okay. Here you can see bar plot without colors. So at the bottom of each bar plot, you have the active fraction of the DNA viruses and the RNA viruses. So we were able to identify, trust me, we were able to identify between 11 and 30% of active DNA viruses. So here, and for the RNA one, we identified between 3 and 14% of active viruses. So based on the bar plot, we also found that the number of active DNA, RNA, the DNA and RNA phages increased gradually over a month, with a peak of activity in May and June, especially in June, which is just after the snow melt. And these results are very consistent with what the team from the lab, the LBL lab found. So very consistent with the microbial biomass and the taxonomic succession just after the snow melt in May. So if you remember, I said to you before that the DNA and the RNA viral community were both dynamics over season. So we wanted to look at only the active fraction of these both communities. So here, this is the same NMDS than here, but only for the active fractions. And as you can see, the active DNA phages appear very dynamic compared to the RNA ones. And because there were highly dynamics, we were able to identify the 4 distinct clusters based on the sampling season. So to conclude, we had in our position a very unique data set to study the diversity and the activity of both DNA and RNA viral communities. We found a very abundant and unexpected RNA viral community. We also find that the phages dominated the whole viral community in soils, I mean, in East River Watershed soils. And this fraction of active phages were very dynamic over seasons. So if you have to remember one thing to my talk today, it's that phages could play a very key role on mountainous soil ecosystem functioning through the interaction with the microbial communities, and all together triggered by the early snowmelt and the climate change. So with that, I would like to thank Simon and the team who collect the cells. Thank you.
Thank you, Clement. We have, I think, one question online we're going to take now, and then if people in the room have questions, we encourage you at the break to find him. Regarding the community dissimilarity of the RNA viruses, it's consistently ranging between 80 and 100%. If they understood correctly, where do you think all of these viruses are coming from, and why aren't they detected at the other months?
Good question. So where are coming from? I would say it's the RNA viral community. What we identified is very linked with the host dynamics. So I didn't show a lot of result about the virus host dynamics today, but for example, I have this bar plot showing the same one for the DNA one, but this is for the RNA one. And so as you can see, the host dynamics, the host is very dynamic over seasons. And for example, here in September, you find a lot of fungi, and it is why we found, I guess, a lot of viruses from infecting or supposed to infecting fungi, and so on. For example, here in June, so just after the snowmelt, you have an increase of plants in soils, and it is why we found some RNA-phages infecting plants. So I think it's really linked to the host dynamics.
All right, great, thank you.